Hello everyone and welcome to this webinar of the Research Working Group. Today we are going to make a brief report about one of the efforts we are doing in one of the objectives of our group, which was the identification and prioritization of different strategies along the different research gaps that we could identify regarding the current practice in pediatric solid organ and hematopoietic stem cell transplantation. My name is Eduardo López-Granados. I'm the clinical immunologist at the Paz University Hospital. I'm the research working group coordinator. So, as you know, in previous meetings and along different board meetings and in the previous activity of the working group, you know that one of their lines of activity is the identification of different research gaps in our current practice in pediatric transplantation that will organize and define future research strategic plans in order to try to fulfill those gaps. So you know that we are doing different efforts trying to identify those gaps, and a good opportunity is the activity that is being carried out in different working groups for our network. For instance, the healthcare or the clinical guidelines and the education working groups. And then the activities of these groups sometimes will help us to identify where those gaps could be, and after efforts done, for instance, in the healthcare working group, in order to develop and carry out a clinical audit, a multi-center clinical audit, about the current protocols and procedures in the detection of a very common or a common complication in any type of transplant, not only solid organ, but hematopoietic stem cell transplantation, which is the development of post-transplant lymphoproliferative diseases. As you know, the healthcare working group have been for a while just conducting a clinical audit, which has identify the current practice in terms of the early diagnosis and management of PTLD, the post-transplant lymphoproliferative disease. And then this audit just raised a lot of interest in our network, and we are working we're preparing a paper, a manuscript to publish the conclusions of this effort. And then one way in which we decided we thought that we could identify potential research areas of interest would be the fact that by the analysis of the clinical audit or clinical guidelines in different areas, we could identify what is missing, what is lacking, or how we could improve our clinical audit, our clinical practice by the advance of different research areas that could improve our tools and our resources to perform better our clinical management. So many of the conclusions of this working group in the area of PTLD, the healthcare working group, is that we could improve our management of PTLD by the inclusions of new biomarkers that could help in a day-to-day basis to follow up the evolution of chronic viral infections, mostly Epstein virus infection, and how we could introduce better and reliable biomarkers that could help the awareness and the clinical decision in terms of the management of PTLD. So with this idea in mind, among the different areas that we explored in a previous effort that was shared with you in a previous webinar, you know that what we did is just in different areas that had been identified as potentially relevant for research efforts. We already identified one that could be important to address in this area of PTLD, and together with the conclusions of the clinical audit, we decided that in terms of making an effort within the research working group, we could find some areas in which the literature could be initially easy to analyze in terms of the number of manuscripts to be reviewed, And then, because of the priority that was established by the healthcare working group, we decided that one area in which we could try to make an analysis of the literature could be the biomarkers that are currently in development or just which are promising for the monitoring or immune monitoring of latent and chronic viral infections in order to identify the risk of PTLD. After doing an initial search, we just identify potential reviews that could address this area in the medical literature. And then, after making just a simple analysis by database search, we decided, and we explained in a previous webinar, that we were to address this effort by using adhering to a current methodology. We decided to use one of the approaches that could be what Cochrane proposes as an overview of reviews. So this kind of methodology will be addressed just to extract data from the different reviews that are published in the literature, and we follow this methodology in order to this effort. Then how we did this was to initially make a review of the literature, as I said, to identify potential gaps in this topic. In concrete, just more specifically, it would be biomarkers for monitoring or immune monitoring of latent or chronic viral infections, post-transplant lymphoproliferative disease risk, both in solid organ transplantation and hematopoietic stem cell transplantation. So initially, although just by doing this literature search, we identify how the literature about PTLD is quite sparse, but there are many cases and reports, but there are not many reviews, and we decided to focus on reviews, either systematic or general, that could address the effect of biomarkers and how those biomarkers could improve the monitoring of PTLD to find potentially relevant gaps in this area that could be addressed in future research efforts. So we recognize that although ERN TransplantChild focus, as you know, in pediatric transplantation, it is possible that some of the reviews that we included in this analysis report results that are only obtained in adult transplantation. Then the research gap could be not in the identification of potential biomarkers in themselves in the PTLD area, but in those biomarkers that could have been identified, validated, or developed for routine introduction in adults, but still the gap will be then the replication or adaptation of the information that has been previously validated in adults in the children population of transplanted patients. In order to conduct the search, we searched in different databases. For instance, the Cochrane Central Register of Control Trials in the Cochrane Library, Mainline, and some others. And for a period of time of 10 years, from 2010 to 2020, and we included reviews of studies, including either systematic reviews or interventional or general studies. With this strategy, we identify a number of 84 publications that we found pertaining to monitoring of biomarkers for PTLD. And then we use this with a combination of words that included post-transplant lymphoproliferative disease with immunosuppression, cellular, molecular, omics, biomarkers, signatures. And then when we identify these 84 reviews, we apply a methodology for the selection of studies. So at least 2 review authors have independently screened the titles of these manuscripts, of these papers, and have assessed the filtered reviews for potentially eligible studies. If there was no consensus among these 2 reviewers, a third one would have solved the disagreement. And then once those studies were identified for data extraction and management, we have used a standard form to extract data from the reviews that all the reviewers use. And then this consisted in the following fields. We tried to identify first the type of study, then the design and objectives of the study, then the type and number of studies that were included in the revisions, the study population in terms of age, number of patients, and type of transplant, the description, and this is very important, of the type of biomarkers that were reflected in those papers, either based on DNA, RNA, protein, cells, or others, the main results of the studies, the conclusions, and the discrepancies of the review. Then one review author extracted the data, and another just checked independently if the extraction had been properly done. And then if there were discrepancies, a third author was involved in the extraction of data if this was necessary. So the very preliminary results that we will expand in hopefully a publication that we will try to conduct from the research working group is that as it cannot be in another way, because this is still an area that is very relevant in terms of clinical management and in terms of prognosis and even mortality, still in the field of pediatric transplantation. There is a lot of interest in the literature in developing biomarkers in order to improve and achieve a more personalized approach to the diagnosis, management, and treatment of EBV, mostly related PTLD. So general biomarkers that could measure the extension of immunosuppression may have a role in avoiding PTLD. As you know, there is a very strong link between the level of immunosuppression and the risk of PTLD, and still just tapering or just adjusting immunosuppression is one of the therapeutic strategies that is employed in the initial approach to the management of PTLD. So biomarkers for immunosuppression, as with any other post-transplant complication, are very important in order to avoid the risk or decrease the risk of PTLD. And then we decided to go more specifically through the different biomarkers that could predict the risk of PTLD development. And then it was important to exclude within this initial collection of papers those that were not devoted or focused on procedures, those that were focused in procedures other than hematopoietic stem cell transplantation and solid organ transplantation, immunosuppression that was not related, or immunosuppressed status that were not related with transplantation, and then reviews about T-cell therapy that were not mentioning specifically transplantation, others in terms of language, they were not written in this, and for any other reasons from those 84 that were originally considered for the review, we finally selected 55 for full text review. And then these groups were divided after the extraction of data into different groups, 28 that were literature reviews, either narrative, systematic, or meta-analysis, and 25 that were general reviews focused more on the actualization of the topic and reporting the state of the art and reflecting the trending for future research initiatives. And those will be specifically depicted in future deliverables as the full report of the manuscript that we will try to make public from this effort. And then as a general conclusions, the reality is that the reviews that we analyze mostly involve adult transplantation. Some of them or many of them include mixed populations, children and adults together, but there is little representation of transplanted children in those that report mixed populations. And we only found 2 reviews that are specifically focused on pediatric transplantation. And this, as you know, is a general observation in many of our efforts when we talk about the future research initiatives in research transplantation, and is again a reflection of even the intrinsic nature of the ERN TransplantChild, which is to understand that pediatric transplantation requires, in comparison with adults, a particular attention because of the lack of many evidence in this area. Regarding the type of transplantation, we identify in terms of solid organ transplantation 9 reviews that consider any kind of transplant, and this is in line with which is the view of TransplantChild as a transversal approach to all types of transplantation, and those includes either solid organ or hematopoietic stem cell transplantation, but many of them, 25 out of 28, imply or considering studies implying solid organ transplantation. 9 of them they were mono-organ, although the majority likely of studies pull together any kind of solid organ transplantation. Although, just considering the number of transplants performed, they were mostly focused on kidney transplantation with liver and heart, following the frequency of those types of transplants that were included in the review. Hematopoietic stem cell transplantation is less represented in this group of reviews, and approximately half of them include this type of transplant. And 3 studies had a small number of patients, and there is also one that is focused more in nurses' practice than in medical approaches. We also observed that the aim of these reviews were mainly focused on therapies and diagnosis, and none of them assessed specifically the role of biomarkers as prognostic factors. But in most of them, biomarkers are mentioned either directly or indirectly. There are 3 reviews that did not consider implicitly biomarkers, as they were more focused on imaging methods that could help the diagnosis of PTLD, or either were missing data that would preclude us to include or to extract the information about biomarkers. Monitoring EBV status has been definitely an important component of post-transplant care in any kind of transplantation, as you know, and this is also a conclusion that was taken by the clinical audit that we conducted among the different hospitals that participated in the study in our clinical in our healthcare working group. And most of the reviews analyze the status pre-transplantation, either seropositivity or seronegativity of the receptor and the donor, and then in post-transplantation to determine the risk of disease. However, the specific details about the analytical methods that were employed for this monitoring were not reported in many of the reviews, and then it is not easy to detect or to conclude how EBV viral load is detected, how it's measured in peripheral blood by any of the techniques, mostly amplification by PCR, or determined by other type of approaches. So the diagnosis, the management, and these are the conclusions of this preliminary analysis that we just shared with you today. The conclusions is that the diagnosis, management, and treatment of both EBV positive and EBV negative PTLD remain a clinical challenge. At present, there is a need for non-invasive, specific, and sensitive biomarkers that can contribute to improve clinical care. And in the field report, we will go through the status of different biomarkers that are either in validation or in clinical identification, just for clinical validation, and how we could speed up the potential inclusion in the clinical practice in the close future of these biomarkers. And then it's also a conclusion that the research is to identify new biomarkers is being focused primarily on molecules of the viral, immune, or lymphoma region that can be detected in the blood. So again, in order to be a reliable biomarker and to perform frequently and on the follow-up of the patients, obviously blood as the specimen that in which this analysis should be done is the most suitable specimen, as I said, in order to perform this frequently and without not being very invasive for the patient. Although there are several promising candidate molecules that could be included in the clinical practice, there are still significant challenges when identifying biomarkers in terms of how they could really be a specific indicator of risks for the development of PTLD. and the diagnosis and the response to treatment. And this is related with the standardization of how these biomarkers should be identified and what would be the limits in order to predict the cutoff values, for instance, to predict risk, and how to link the specific evolution of the viral infection and the immunosuppress or the immune status in order to be able to really predict in which situation the immunosuppression should be excessive in order to facilitate the initiation of the PTLD process. And then it will be important to combine, as I said before, the analysis of candidate molecules with measurements of the viral load, so that good correlation of viral load and molecules that could reflect immune status would be important. And as a conclusion, many of the studies that are so far identifying promising biomarkers have been conducted in comparatively small numbers of adult subjects. So what will be the proposal for the future as a network in terms of proposing areas in which we could work together and just establish a good platform for future research efforts? We believe that after the analysis of all this area and the identification of gaps, it will be important to establish multi-center pediatric studies with a large patient population to increase the cohort size. And then it will be important with this effort really to include and evaluate promising biomarkers that could be validated and just evolve them to be informative biomarkers that could be potentially included in. And with this, I just thanks for your attention, and I just I invite you to future either webinars or either lecture of different of the complete results that we will put together and hopefully publish, or just speak more specifically in future webinars with more specific details about the data that was identified after this revision. And with this, I just want to thank you for the attention, and I'll see you in the next webinar. Thank you very much.